All right, so what I have set up here is the complete setup for how we're going to do this enzyme lab. So there's the schematics here in your lab manual, and just to go over some of these components, let's take a look at this interesting tube, which is actually just a regular burette. And so most often in chemistry lab, you would have used it this way, where you can open or close it, and then maybe you're doing a titration or something. But in our lab, we're actually going to use it upside down. And so that's what I clamped in here. And I also filled it with water, and we'll show you how the technique for how to do that later. But right now it's full with water, and probably the hardest part of this lab is to read this thing upside down. So whoever is going to be the person that is reading it probably should grab a piece of paper too to help kind of seeing the tick marks. But when the person that's the timer is yelling out now, the person that's reading is going to have to orient themselves and see where is the level of water. And so, of course, now 50 is up here and 0 is down there. And so, for example, if the level was here, that's actually 47.5. And then this would be 48, and then so forth. So just take a moment and get really comfortable reading this, because it is a little bit stressful when you're going through the experiment. But keep in mind, each trial is super fast. If you do mess up, you can just redo it again. So back to our setup. So what we have here is a water bath. We filled it kind of halfway up with just plain tap water. And in this case, I have filled this one, and we're going to zero that later. But before that, we're going to take a look at some of the other equipment. And so what we have here is just a Erlenmeyer flask with a stopper. It has 2 glass tubes and 2 hoses here. The shorter hose you're going to use for your syringe. And so this syringe has milliliter marks on it, and this is where you're going to do your dilution according to this table down here. So each trial is going down a column, and it tells you how much water and how much hydrogen peroxide you need. And so when you're doing this, it's essential that you start with the water first, and then move the syringe over to the hydrogen peroxide, just so you're not cross-contaminating and messing with the dilution or the concentration. The other thing that I think is pretty clear, if you just take a look at these 2 solutions, they both look like water, which means that you need to be super organized and label your containers. Even with these bottles, it can happen that you go over to the counter and pick them up, and a student in the previous section might have put the wrong cap on, or it's just not the right thing. So being super organized, keep it labeled. You can use the syringe directly into this. However, when it starts getting low, it's hard to get in. If you do pour it over into a beaker, then of course label your beaker so it's easy for everyone in your group to see which fluid is what, and that makes it easier for that. So then after you make your dilution, you're going to stick the syringe on the shorter tube. Someone else in your group has probably been nice enough to go over and get the enzyme extract that we made earlier. And you're going to have the same amount of enzyme for each trial. So it's always going to be 10 ml, which means that in this experiment, we're keeping the catalase concentration constant. What we're varying is obviously the concentration of hydrogen peroxide for each of the trials in the experiment. So once we're done with that, we're going to seal this tightly. You have your trial dilution ready-made in this. And then once you squirt the hydrogen peroxide down to the catalase will help out with that reaction that is going to lead to water and oxygen. And the oxygen is a gas, which is of course going to want to escape and bubble out of this tube. So what we're going to do is we're going to immerse this before we start the experiment and stick it up into the burette. And then it's kind of key that you also make sure that you don't have it accidentally slip out during the experiment. But if it does, remember, this is a super fast experiment. You can just redo it, the trial. So during the experiment, one person is going to agitate this. We keep the reaction going and everything is mixed nicely. So then gases are going to form, or oxygen is going to form and start bubbling here, displacing the water. And then the person that's reading the graduated cylinder will yell out the numbers, and another person will record it into the lab book. So it's key that you have made a table so it's quick to record times and volumes as it goes along, because things will happen quickly. And then new for this time, we haven't done this before, but we have experienced sometimes that we get enormous bubbles traveling up, doing a major displacement. And so this time we're going to actually try putting some of this gauze or cheesecloth that has large holes in it. We're going to try to break up those big bubbles by putting this on like this and insert that into a burette, and hopefully get a little bit more finer bubbles, so it makes it easier to read the various readings.
Cool. All right, so I'm going to show how you set things up for the beginning of each trial. To start, we have already, my lab mate has placed 10 mils of our diluted enzyme solution here in our little Erlenmeyer flask. Make sure before each trial you actually kind of clean this off, do like a rinse with distilled water, so you have a kind of clean, fresh enzyme each time. And then I'm going to need to have my appropriate dilution of my substrate, which I'm going to put in my little syringe here. To illustrate, I'll show, for instance, trial 2. Trial 2 is going to be 2 mils of hydrogen peroxide, 3 mils of the distilled water. In each case, we make sure there's a constant volume. Again, another source of variability in this reaction is the total volume that the reaction is taking place in. So we want to make sure that each trial has the same volume of liquid. So, as you heard, we want to make sure we start by bringing up our water. So I'm going to look here. I need 3 mils of water. All right, so I am going to suck up my water here. Again, I need 3 mils, so I actually suck in a little more than 3 mils. And then you can knock out all the air bubbles. And you bring the plunger up until the black of the plunger is exactly on 3 of the marking. That means there's 3 mils that are going to get squirted out of water. And then for the hydrogen peroxide, the 2 mils, we just have to dip the nose into the hydrogen peroxide here and continue sucking up until I get exactly to 5 mils on my marking. And there I go. So there's 5 mils of liquid in here, 2 of the hydrogen peroxide, 3 of the water, so that's my dilution for trial 2. This then gets very firmly attached onto the short tube of this little setup going into the stopper. You really want to make sure that this is sealed well and isn't going to fall off when you're going through the experiment. Here we have our 10 mils of the enzyme solution, which is constant from trial to trial. I also want to make sure I have a really good seal between my rubber stopper and the flask. So I'll use some water on the stopper before I put it in. Then I can put the stopper in and really seat it very, very firmly. Obviously don't push it so you're going to break anything, but you really want to have it firmly in there. And even though it's pretty darn low, you will be able to kind of rock it and get it out when you're done to do the next trial. So now we're kind of set here. When I want the experiment to start, I just squirt the substrate into the enzyme. The reaction will happen. Any gas, oxygen that gets produced will exit through this hose. So this hose now is going to have to be placed in the burette where I can actually capture and measure the amount of oxygen that's getting created. So I'm going to basically reach under here and put it here. I want to make sure it goes far enough up that it doesn't fall out as we do the actual experiment. And so what we're going to do in a few moments is show you the actual experiment in progress.